First of all, thank you very much for the invite here and the opportunity to come and present our data. As you mentioned, I'm from King's College London, and I'm going to talk about some of the work that we've done on genomic repositioning, with particular emphasis on the dementias and Alzheimer's. Here's my statement and declaration, which of course is mandatory. And here's just a brief summary of my lab interests. So really, it probably could be best summarized by we're interested in biomarker identification across a number of different processes. That would be summarized by neurodegeneration, but also regeneration and pain. And I'm just going to talk today about one aspect of that, which is shown up here on the left, which is drug repurposing in the field of Alzheimer's disease. So I'm going to give a very brief introduction to Alzheimer's, some of the problems about finding drugs there, and one of the solutions that we've taken using a genomic approach. So I'm sure you're all very familiar with Alzheimer's disease, but I'll just go over some of the key facts to set the scene. So approximately, there are a minimum of about 35 million people worldwide with dementia. That's clearly an underestimate. And it's been predicted that by 2040, that will at least double, although some estimates have it at quadrupling. Of those dementias, then greater than 60% of dementias would be classified as being Alzheimer's disease. And this has a huge global human and financial cost, which at the minute is estimated around $600 million and set to rise as we're an aging population. I think one of the key things to say about Alzheimer's is if we look at the state of drugs that are available at the moment, then we would say that they're symptomatic treatments. They're not disease modifying, but they do have important benefits. But really what the field needs in Alzheimer's and what healthcare needs are disease-modifying drugs, things that actually slow the process down or halt it, or ideally, of course, reverse it, but that's a very difficult problem. If we look at Alzheimer's disease, actually there are only really 5 major drugs that have been you can get prescribed now, and they fall into 2 very simple categories. So 4 of them are cholinesterase inhibitors, and simply they make a neurotransmitter stay around longer. So the acetylcholine is depleted in the Alzheimer's brain. The cholinesterase inhibitors allow more acetylcholine to be around there and improve brain function. And also we have an NMDA antagonist, which is to reduce glutamate signaling and exocyticity. So again, they're symptomatic, they're not disease modifying. It's a little bit strange that there are only 5 licensed drugs for this when you consider the range of biological targets that we have in AD. And this slide just summarizes some of the major problems that go around in AD. Of course, there's the polyproteinopathies. These are the A-beta, the neurofibrillary tangles, which are shown in the little image on the right there, and the alpha-synuclein. Probably one of the biggest characteristics anatomically that you're familiar with is a specific neuronal loss, for example, hippocampal cells, which underpins the loss in memory, but also as well, it's probably better described as a synaptodendritic rarefaction. So not so much the loss of the cell bodies, but the loss of the neuronal processes that send and receive information. Of course, there are many other aspects to it: mitochondrial dysfunction, multiple transmitter defects, as I've mentioned, acetylcholine and glutamate, aberrant neuronal network activity, something that isn't particularly well covered. You do get lots of extra activity. For example, you see a lot higher incidence of epilepsy in AD. But there's also things like reduced neurogenesis, epigenetic changes, lysosomal changes, dysregulation of intracellular calcium levels, oxidative damage, and cell-to-cell spread. I think the take-home message from this is that there are a lot of targets there for the drug industry. So why are there very few drugs that have been found to have any impact on the progression of Alzheimer's? And in a way, the answer probably is in that slide as well. One of the reasons why it's difficult to find this drug is that very much Alzheimer's is a disease of co-pathogenicity. Lots of different factors interacting together, so it's very hard to find one single drug that will ameliorate any one of those pathways and have an impact clinically. I'm sure you're all very familiar with this slide, but we put it up there as justification, largely when we're applying for grants. This is a summary of the drug discovery process. On the top panel here, we have the hit to lead diagram going through all the clinical trials. And of course, you know that we start with a lot of hits, relatively few leads, and even less of those make it through to phase 3. In fact, for AD, there's about a 98 to 99% failure rate. The bottom panel also shows the number of new molecular entities that are approved by the FDA. It's a little bit old now, it's 2013. There's actually a lot less than I think most people think. There's only about 1,400 are approved. So there isn't very many drugs, they're very expensive, and just if you look at the bottom line there, again about 2012, if we look at the number of clinical trials, then cancer gets a lot of emphasis. There are about 1,700 clinical trials going on in that year, but only about 30 clinical trials for Alzheimer's disease. So it's a very important disorder, it's very difficult to drug, and there's very few clinical trials that are going on to try and address this problem. So this comes more to what we're doing with genomics. Is it possible to use existing drugs to try and develop or identify some disease-modifying candidate drugs in AD? So this is what's commonly known as drug repurposing or repositioning. And essentially what this is doing is finding a new role for an existing drug that's been approved. Of course, it has enormous advantages. You know, the dosing, the tolerability, and also much less cost because you don't have to drive them through the same clinical trials process. Drug repurposing can largely be divided into two major categories. Of course, it's much more complicated than that, but we can have it as an on-target approach or an off-target approach. An on-target approach would be where an existing licensed drug is interacting with a pathway, just perhaps in a different part of the body or a different organ. There is, of course, a classic for that. So sildenafil, which is a specific phosphodiesterase inhibitor, was originally targeted for angina, a blood pressure-related disorder, and of course, it was far more successfully repositioned for male erectile dysfunction. So that's, of course, Viagra. But there are also off-target examples, and this is where you have a drug that interacts with one pathway, but then has a side effect and does something else that is of potential clinical benefit. There's lots of examples of this. Aspirin is perhaps the most common, but given that we're working on dementias, I've just shown one here for amantadine. This is licensed for influenza, but actually was discovered to be an NMDA receptor antagonist, which was licensed for use in Parkinson's disease. This is very much, again, about making neurotransmitters hang around a bit longer. So what is our aspect of the drug repurposing study? Well, we're taking a genomic approach, and it's based upon the connectivity map, which was initially published by the Broad Institute. And this is very much an off-target approach. This slide is, in 2 slides, I'm going to try and summarize the connectivity map, but I'm more than happy to talk to anyone later if they want any more details about it. This is about diseases or disorders that have associated changes in gene expression. So the very easiest way to think about this, particularly with reference to Alzheimer's disease, if you have that disease or disorder and it has changes in gene expression, we can call that the gene expression disease signature. Now, you can generate that signature any which way you like. Think about this maybe as 500 genes that are upregulated and 500 genes that are downregulated if you have AD. You can generate that signature via microarray, via NGS, via GWAS, as we've just heard. You can do it manually or by metadata. There's lots of different ways to generate your disease signature. Effectively, it's just a set of gene expression changes that define your disease. And then the CMAP really relies on finding drugs that will reverse that gene expression change signature. So in AD, if you have 500 genes up and 500 genes down, the connectivity map is finding a drug that will just simply switch that set of genes around. This slide is actually from the original article on the connectivity map, And if you look on the left of the slide, then we have our biological state of interest. Think about that as Alzheimer's disease, and you can use a microarray approach, if you like, to define the genes that are up and down regulated. And then they've taken the panel of approximately 1,300 FDA regulated compounds and passed that over some MCF7 cancer cells and individually defined the set of genes that each FDA approved drug will regulate. And that then builds a drug profile database. So in the very simplest case, if we look and we have a disease state like Alzheimer's, we just try to find a drug that will anti-correlate with those gene expression changes. It will change the way the genes are expressed. If it's upregulated in Alzheimer's, the drug will hopefully downregulate it. So it's very much a correlative approach. And as mentioned, this was all done in MCF cells. So it had a cancer focus. I mean, that sounds very good. Does it work? Well, this is some of the proof of concept data. The brief answer is that yes, you can get this process to work and identify compounds that will reverse the changes. This is one of the examples, one of the early examples that were published. We have a human lung adenocarcinoma signature that was generated, run it over the CMAP drug profiles. It identified drugs, cimetidine, that would potentially reverse those changes. If you put that drug back into a mouse model of that, then you can see on the left you do get shrinkage of the tumour. So that's good proof of concept that it might work. Well, we wanted to take a very similar approach, but for Alzheimer's disease. Now, clearly, Alzheimer's disease is a neuronal-based disease, and we were applying a repurposing strategy that was genomic, but also had a cancer focus. So we wanted to shift that back to a more neuronal aspect. So we started the Smart AD initiative, and we are going to try and repeat the CMAP, but rather than using cancer cells to develop the drug profiles, we're going to use human iPSC cells that have been differentiated towards a cortical phenotype. For our disease signature, I won't go into this, but again, I'm happy to talk about it later. We actually use the public databases and the metadata to identify a consensus disease signature of AD that represented early AD as classified by clinicians. What happens when you run your AD signature over, this time starting with the CMAP database, can you find any drugs that will anti-correlate with that signature? And this shows a distribution of our hits, and the answer is yes, you do. You find good set of correlations for drugs that will drive the gene expression changes in an opposite direction. So these would be candidates for your repurposed drugs. Without going into too many details, one of the nice things about this approach, when you look at the complexity of what those drugs are, they fall into lots of different classes, which is exactly what you want in a drug discovery pipeline. As I mentioned here, the original CMAP was done in the human MCF7 breast cancer cell line, but we're going to repeat it here in the panel labeled NMAP in a human iPSC cell line, and I'll give you some summary details of what we do with that next. Just a very, very brief bit of proof of concept data here. This is where we've taken some FDA-approved drugs and just passed them over human iPSC cells in triplicate and shown that you can get a reproducible change in the gene expression profile of those human iPSC cells. You can see the PCA plot on the right, and you do get a relatively reproducible profile. So these are gene expression changes that are off-target driven by the FDA-approved drugs. And again, approximately, as you expect with most of these transcriptome experiments, 500 to 600 genes upregulated, 500 to 600 genes significantly downregulated. And really, this slide summarizes the majority of our effort. There are about 1,300 drugs in the CMAP database. When we ran our consensus disease signature over that, we identified that there were approximately 200 drugs which may have a role in reversing the Alzheimer's disease signature. So our first thing was to take a look at what those 200 drugs were, do a systematic review, and take away anything that we thought wouldn't be suitable for actually being used as a treatment. And we were left with about 160 compounds that potentially could reverse the changes in the AD disease signature. And this is where we bring in our NMAP processing. So we took those 160 candidate drugs and passed them over human iPSC cells that have been differentiated to a cortical character and regenerated the drug profile. So the question then is, we know that the drug can drive gene expression changes in cancer cell that would reverse the AD changes, but can it do it in a neuronal cell? You can see as the numbers come down here that actually we had about a 75% attrition rate there. So only about a quarter of our drugs that were identified in the cancer CMAP profiling retained that in the NMAP. So we already were having a much greater specificity. This, of course, was where the genomics aspect of it came in, because when we put our drugs into the NMAP, we chose to do that all on the array platform. It was the same array platform that the original CMAP was developed on to try and hold for the consistency of that. Once we have our 40 candidate drugs, we then switch to the more standard drug discovery process. So we go back into the lab and ask, how do they perform in the standard set of in vitro tests for something like AD? And that's summarized here in this slide. So we take our 40 NMAP identified candidates and pass them through a series of in vitro assays to see, will they lower all of the sort of histopathological markers of AD, which we hope will have some clinical impact. This is just a very brief bit of that primary data showing that, yes, some of those 40 candidates perform extremely well in those in vitro assays. But of course, we decided to have a more stringent approach, and we would only choose drugs that performed well in a number or all of those assays. This is summarized very much here. So our 40 drugs that have an in silico potential for being AD repurposed drugs actually then were whittled down to 12, which pass our in vitro tests. But again, very encouragingly, these drugs were across a number of classes: antibiotics, NSAIDs, receptor antagonists, so on and so forth. So this then is our enriched set of candidates for the AD therapeutics. And then it's very easy, because we've all done this on a genomic processing. You can just have a look back and ask, well, what pathways are those drugs hitting? That's a standard pathway enrichment that we all do. And we can pick drugs that hit different pathways so we can mix and match the potential of our candidates. And this really summarizes where we are now. We had 12 drugs pass our in vitro tests. Again, we did a bit of systematic review on those, sent them out to the pharmacologists. Which ones do you think will be the best to use? Whittled that down to 6, and they're now on the in vivo screen going into the 5x FAD mouse. Now, I'm running a bit short of time here, so I'm not going to mention how we coordinate that with other studies, but suffice to say that we've now got our 6 candidates. They're in the in vivo, and we're in the process of assessing of how they do that. And we coordinate this data with our other genomic repurposing strategies to try and get a sense of how drugs work across different pathways and different platforms. So most of the work I've done here, of course, was spread across lots of different groups and lots of different initiatives. So that's my thank you slide, and thank you very much for listening.